Okay, so yeah, you can start. Great, thanks, Chris. Okay, so hi everyone. So it's my pleasure to basically have you for the last CFAR seminar of this 2020-2021 season. We have Rachel Bender-Ignacio, Ignacio, who's who is our new face speaker seminar, and then we have another followed by Nicole Doria-Rose from the VRC. And as you know, the CFAR seminar provides a forum for presenting and discussing state-of-the-art research finding in HIV pathogenesis, virology, care, and treatment, and to provide an opportunity for ongoing enhanced communication and multidisciplinary collaboration among HIV/AIDS researchers across the 5 CFAR affiliation institutions and their partner organization. So the next CFAR seminar will be in October, and the new line of speaker will be announced in August. And it is my pleasure to have to introduce Rachel. She's an infectious disease physician, scientist, and assistant professor at the University of Washington, where she recently was named the director of the UW AIDS clinical trial units. She completed medical school and an MPH in epidemiology at UW. She trained in internal medicine at Mass General Hospital and Harvard, and was briefly on faculty there prior to returning to UW for infectious diseases fellowship. The majority of her patient care and research has been focused on HIV, co-infection, and malignancy, specifically on the intersection with co-infection, immune activation, and HIV outcome. Rachel is currently director of the HIV Medicine Association Board. And since the beginning of the COVID pandemic, she has also taken on significant leadership on COVID-19 treatment research, including service as the medical director of the COVID-19 Clinical Research Center at the Fred Hutch, and as protocol lead of the International Active-2 study. The work discussed today is the result of a recently completed CFAR New Investigator Award grant in 2018. So it's our pleasure to have you, Rachel, to talk about Mycobacterium, tuberculosis infection, immune activation, and risk of HIV acquisition.
Thank you so much for that really kind introduction, Marie. I don't have a disclosure slide, but I have no disclosures related to this work. So to start off with, I wanted to give a brief introduction about immune activation and the risk of HIV. This is a topic that I've been interested in since about 2015, and is actually the topic of my K Award. And my K Award is really looking at the influence of viral co-infections and HIV risk. And so I had really the great benefit of getting to have a CFAR award to be able to study the influence of Mycobacterium, tuberculosis, and HIV risk as kind of two complementary studies. So what do we know about immune activation and risk of HIV? So some of the best described data is from the STEP vaccine study. So this was a Merck Ad5 vaccine in which people were randomized to vaccine or placebo. It took place in the Americas, including South America and the Caribbean, as well as Australia. And what they really found was that certain subpopulations in this vaccine trial actually had an increased risk of acquiring HIV rather than protection after being vaccinated. And it wasn't due to any HIV antigen specific responses that increased risk of acquiring HIV. It was due to a high mock interferon response. So meaning just T cells stimulated in general, those that secreted more interferon gamma in response to general stimulus. Was associated with risk of acquiring HIV. We also have studies as sort of sub-studies on two other interventional PrEP studies that showed that particular markers of immune activation, in this case, soluble markers of immune activation, IL-2, IL-7, IL-12, TNF-alpha, and IP-10 predicted people who would go on to acquire HIV within, whether or not they were receiving treatment, but in the placebo arms of those studies. And then I think some of the most really provocative data for why we should worry about MTB as a risk factor for HIV comes from some macaque work that was done by Christina Paris and her group, in which they were testing an MTB SIV oxytroph vaccine as well as a BCG vaccine versus placebo in macaques and infant macaques, and then challenged them with SIV. And those that received either of the two vaccines, either the MTB SIV vaccine or the BCG vaccine, were more likely than those that received placebo to acquire HIV. And there was a correlation with having an elevated TNF alpha prior to or subsequent to vaccine administration. So we know that MTB, elicits a lot of interferon responses, and some of these similar responses with, again, a lot of these soluble markers and T cell markers that we know about. And so, we really wanted to answer this question of whether being infected with MTB and having an associated immune response to that infection increased risk of acquiring HIV. So why is this important? So we think that MTB, Mycobacterium tuberculosis, infects approximately a quarter of the whole world. This is previously listed as up to a third of the world's population, but I think the most accurate data is probably closer to 24%. When we talk about latent tuberculosis infection, what we talk about is having either a positive TST or tuberculosis skin test, also known as a PPD test, or one of the newer blood tests, which is an interferon gamma release assay, which is essentially stimulating T cells in the lab with two specific proteins, ESAT6 and CFP10, and looking for secretion of interferon gamma in response to TB antigens. And in order to qualify as having latent TB, one has to have an absence of clinical symptoms, which would qualify somebody as having TB disease. There's also been sort of new recognition that there's a status called incipient TB. And what incipient TB really means is evidence of systemic inflammation or sort of a response that predicts that someone will go on to develop TB. And a lot of those sort of responses that we see that are trying to control TB as it's activating over months or years before it becomes detectable clinically are these sort of interferon type responses that are very similar to what we would be concerned about as predicting a risk of HIV acquisition. And we think that this topic has a lot of public health importance because of currently around the world, there are no countries in which TB and HIV are co-endemic, in which people who are HIV negative are given chemoprophylaxis against tuberculosis for latent infections. So we know here in the United States, among immigrants specifically, or people who've been in the carceral system, we test people with a PPD test, a TST test, or with an IGRA, and treat people to prevent TB disease. But that's not true in the majority of the world where TB is endemic, and especially not in people who are HIV negative. So, if in addition to PrEP, we could use LTBI chemoprophylaxis to prevent HIV, that would be important both to, knock down future risk of TB, but also prevent HIV. So I talked a little bit about the STEP study. We created a nested case control within this study. So this STEP study, which was also called HVTN 502, and there was a follow-on study that extended follow-up for these participants called HVTN 504, had high-risk HIV negative males and females in across the Americas and Australia. And we identified as cases everyone in the study who had acquired HIV during follow-up in either 502 or 504 and had peripheral blood mononuclear cells available, still banked for analysis. And so this was an ancillary study that was approved by the HVTN. We then selected as controls to these cases people who did not acquire HIV during this follow-up. They were matched 2-to-1 by study site so that they're coming from the same sort of general risk level for having TB, and also by vaccine arm, either placebo or having received the active vaccine. And then they were frequency matched for gender as well, but not 1-to-1 matched. And then we discarded any cases that either didn't have available samples or had acquired HIV prior to the first sampling time point. So our lab methods that we use, so this was a combination of work done in the SHA lab and the Han lab, both in Sarid at UW South Lake Union. So flow cytometry was done for MTB specific CD4 cells using ESAT6 and CFP10. So these are the same antigens that are used in an IGRA test. And then with intercellular cytokine staining for 4 cytokines. We also extracted RNA from these PBMCs and did ran transcriptomic profiles. So these were used to create these correlates of risk signatures that have been previously well associated with risk of subsequently acquiring TB disease or correlate with TB responses, like the response 5 or MITSTR4, which can discern between latent and active TB and predict TB success. So in this study, because we didn't have clinically defined LTBI status with PPD or IGRA tests, because those weren't performed in these participants, we used flow cytometry and defined interferon gamma responses at twofold the frequency over controls as being LTBI positive, and then using the polyfunctional cytokine responses specific to those antigens. We also performed COMPASS analysis to be able to use that high dimensionality data in a kind of more logical way. And COMPASS is a data reduction and technique that uses Bayesian prediction, and it was developed by Rafael Guittardo at the Hutch. We also then used conditional logistic regression, and this was work done by Jess Long, who's an epidemiology postdoc that works with us to look at, to evaluate the odds of HIV acquisition, adjusting for all of the different covariates that are associated with risk of acquiring HIV in the parent study. So these are the baseline characteristics of our participants. They're relatively young. Most of the cases, and therefore the controls, came from the US and Canada, with a smaller proportion from South America, mainly Peru and Brazil, some from the DR and Haiti, kind of a wide representation in race and ethnicity, and then appropriately proportioned between the vaccine and placebo arms of the trial. If you see here, this risk score as a behavioral risk score that was validated by the HVTN and is appropriately predictive of who will acquire HIV based on things like condomless anal intercourse and number of sex partners. And then we had HIV or HSV serostatus and circumcision for male participants only. So maybe the biggest part of the punchline is that when we just looked at LTBI, we did not find that LTBI itself was a risk factor for HIV acquisition. The results were essentially the same between cases and controls overall. When we then looked at COMPASS scores by outcome, so again, we've got functional scores and polyfunctional scores, which look at the kind of combinatorial different cytokine expression of that's antigen specific, MTB antigen specific. This first graph is just a check to show that this works and we have good correlation. So you can see that in the light blue boxes, those who were LTBI positive had much higher functional and polyfunctional scores, with the functional score being more sensitive for picking up LTBI because it doesn't require having a kind of a polyfunctional response. But we've got good separation between people that we classified as LTBI positive and negative. But then when we look at cases and controls with both the functional and polyfunctional score, we really see no difference here. So this is telling us that cellular immune activation in response to LTBI does not predict HIV acquisition. We also looked at this as a COMPASS heat map. So this is one of the ways that you can look at COMPASS data specifically. And down at the bottom, these heat maps are a little bit difficult to read and interpret, but the different cytokines and the combinations thereof are listed at the bottom. And if you look at the controls stacked on top in lime and the cases in purple, we really see no difference in the pattern of polyfunctional responses between cases and controls. These are our unadjusted and adjusted odds ratio. So again, putting into the equation, it's a conditional regression. So it is matched on treatment arm and country, and then we included gender, risk score, and age. And we can see that this is completely, the adjusted odds ratios are close to 1 and they're not statistically significant. When we did a gender restricted analysis, just looking at male participants so that we could add in circumcision and HSV to serostatus, the results were essentially the same. So then moving on to the correlates of risk score, which I think is the other, because the first part was essentially a negative study, this is the more interesting part. So we chose as our primary exposure of risk as the risk 6 score, which includes 6 genes. Again, these genes are not specific to people who are TB infected, but the presence, the differential regulation of these human genes can predict the development of clinical TB, even including people who are not known to be LTBI positive. We also used for exploratory analysis, because we were able to run these on a Fluidigm plate with as many primers as we wanted. We included Suleiman 4, Sweeney 3, Metzdorf 4, and Response 5, which are all different correlates of risk scores, again, that use differential gene expression to correlate with or to predict different TB outcomes. And risk 6, Suleiman and Sweeney are all interferon associated pathways. So nothing incredibly stands out here when we just look at cases versus controls compared to these different risk scores. We log transformed the ones that were not normal and that were distributed between a scores of 0 to 1 so that we could actually, get a better distribution here. We see that they're all fairly similar, except for some signal in Sweeney 3. And then when we look at specifically the multivariate, so adjusted estimates, we see that actually Sweeney 3 is actually quite predictive of the risk of acquiring HIV, even when including all of the known risk factors for HIV, and including, and actually this result becomes more strong when we look at just male participants and include HSV into the model. We also see that the response 5 score seems to actually be protective of HIV or of HIV acquisition, but only when controlling. It's sort of borderline statistically significant in univariate analysis, but it becomes more strongly protective when we control for the other factors. So because we found no result overall and no result from looking at LTBI itself, we thought, well, maybe these correlates of risk scores are actually picking up either interferon associated or other sort of differential immune responses that are different between people who have LTBI versus not. And this is exactly what we found, although I think the results are maybe even more thought-provoking and didn't entirely answer the question, I think, that we plan to set out, in the sense that you can see that there's even differential directions of effect between in risk 6 and Suleiman 4. Sweeney is not statistically significant in this LTBI group for many of these, but differential directions of effect. Again, the LTBI group, because it is about 20% of the overall cohort, was likely underpowered to really be able to do this stratified analysis well. But I think that the LTBI negative group is really the most interesting. So, I think the way I would interpret this is that if the core scores can predict HIV acquisition, but LTBI status and cellular immune responses associated with TB antigen specificity are not associated with HIV acquisition, then there must be an unmeasured confounder. There must be some other immune provocateur that we're picking up with these transcriptomic profiles that we're not seeing, that we can't measure with our other covariates. So we did find that the Metzdorf score was associated with being a vaccinee, so it was higher in people who'd received the vaccine. We didn't find that any of these scores were associated with gender, the behavioral risk score, or the adenovirus titer prior to vaccination. There was a trend towards differential risk 6 score, Sweeney score, and response 5 by HSV serostatus. So again, that's restricted to males in the cohort. And the scores actually varied widely by country of residence, which is expected given all of the other different environmental, parasitic, et cetera, exposures that people may have living in different countries. So the other thing that's really interesting to note is that when we looked at just men only in this cohort and included HSV2 and circumcision into the model, most of the adjusted odds ratios moved more towards the null, but HSV was retained in the model as a strong predictor of HIV. So I think, one of the take-home points is that these markers are not necessarily picking up HSV infection, but they're picking up something, and HSV is still really important as a risk factor for HIV acquisition, which we already have known through several studies, including some done here by colleagues at the UW. So limitations of this study was that we had a single PBMC sample from each participant. In some of the cases, the PBMCs were greater than a year prior to HIV diagnosis, and that MTB status potentially could change. People could acquire TB in TB endemic countries, and so could convert or actually revert their status. There are probably other unmeasured exposures as we saw from the stratified analysis. With the wide interval between study visits, we weren't necessarily able to look at these predictors, the lab work. We weren't able to necessarily zero it in right prior to HIV acquisition during the time in which somebody may have been exposed to HIV. We also didn't use the clinically approved sort of methods of diagnosing LTBI, but we used, a very rigorously and scientifically accepted method to do this via the antigen-specific flow cytometry, which arguably gives more data than an IGRA or a TST. So how does this tie into the other work that I'm doing? I'm just giving a little tidbit of other work that's under revision, was presented at CROI. So this is a completely separate cohort, the Sabes cohort from Peru, which was a case control study of people who did and didn't acquire HIV. And what I wanted to show was that with this study had 2 time points, so baseline and enrollment, and then a time point just prior to HIV acquisition. And what we can see is that when we enrolled people, many of their cytokines between cases, I left off the key for this, I think, I'm sorry, that cases are purple and controls are green, that there's actually really different levels of soluble markers of immune activation between people who later went on to acquire or not acquire HIV. And then when we looked at these longitudinal plots, these are Tisney plots. So taking all of the biomarkers together, we can see that at the time of enrollment, cases actually really segregate themselves differently from controls. So we can see here that, immune activation does predict HIV acquisition. And I was hoping that TB would be maybe the biological underpinning that could explain this, but no dice. So my conclusions are that there's no evidence that LTBI or the antigen-specific immune responses increase the risk of HIV acquisition. This was done in a population at high risk for HIV within countries with a wide array or wide range of TB burden. But that our exploratory analysis did demonstrate that these transcriptomic signatures can predict HIV above and beyond the known clinical and behavioral risk factors. And so I don't think that using a transcriptomic score would ever replace just talking to a patient and asking them their behavioral risk factors or other things that might encourage us to prescribe PrEP. But I do think that they could potentially be useful to build a purpose-built, so do something like RNA-seq to find a more purpose-built HIV core or HIV correlates of risk signature that could be used potentially within vaccine studies to stratify people into different risk categories, and then probably importantly to not repeat what happened in STEP, to be able to look at HIV vaccine responses that could potentially be risky in and of themselves, like the vaccine that was tested in that study. So I wanted to end with some gratitude to all the people involved in this. So first of all, to all the participants who gave their time to participate in that study and gave their blood and permission for us to use it at a later date, which we did. I wanted to thank the HOPE group, which is my home research group at VID in the Hutch, who I mostly haven't seen in a very long time, but specifically Jess Long, who did much of this analysis, and Ann Dorr, who's been a fantastic mentor to me for a long time and was also on the protocol team of the STEP study. And we did this project with a lot of collaboration from the HVTN, including statistical input from Holly Janes, who works on the biomarkers for the parent study, folks from SATV who did first worked on these correlative risk signatures, and of course, Tom Hahn and Javed Shah and all of the amazing folks in their labs who actually did the lab work for this. So with that, I'll stop and happy to take questions.
Great, thanks, Rachel. So there is one question in the Q&A, so I'm going to start with that. I don't know if you can see them, but so the first question is, which cytokines or how many were used for the polyfunctionality analysis?
Good question, April. I will speed back there. Is my pointer available on the screen?
Yes.
Okay, perfect. So IL-17, TNF-alpha, IL-2, and interferon gamma. So these are listed at the bottom here and the different subset combinations. So these are single responses to each of those cytokines in light blue, and then in dark blue are sort of duet responses, and then triplet, so multiple expressing cytokines here at the bottom.
Thanks. I don't see any additional questions yet. I don't know if some people want to, put them in the Q&A, raise their hand, or which I think you can, although I'm not entirely sure. Rachel. Oh, so Florian. Okay, so there's a few more questions. People raise hands. So Florian raised his hand. So maybe Florian, you can go ahead.
Yeah, just a very quick question. Great talk, Rachel. Good to see you, and
Thanks, Florian.
Very interesting work. Do you have a chance with your transcriptome data also look at SNPs? I mean, obviously not in promoter region, but in the exon areas. Did you look at that also?
So these were done as on a FluidM platform with specific primers. So it wasn't, I'm sorry if I'm making anybody dizzy with the slide really quickly, but there actually is there are a total of 48 primer probes in this set. And we only included in the analysis ones that belong to one of these 5 cores, but they're all ones that have been previously associated with a TB correlative risk signature. And so I think the future steps would be to do something with, RNA-seq. We did also save DNA. I know that's something that Javed's lab has, been really interested in looking at, is polymorphisms in different responses. And so I think there's a wealth of data here if folks are interested in talking more about it in the future.
Thanks.
Okay, so there's time for one more question. Is it possible, given data, to look at association between cytokine levels and viral load levels? And do you think this would show anything? It's from Daniel Reeves.
That's a great question. It's almost like you're reading the paper that we have, in preparation, and not in this cohort, but in the SAP-based cohort. So related to the other data that I showed, and that is in process. And also looks at the timing of HIV, the estimated date of infection to the time of sampling, all the work, Dan, that we did together about looking at sort of timing of ART initiation and acute infection. So that is pending. But again, for the SAB-based cohort, not for the sub-study.
Great. So thank you again very much, Rachel. I think we're perfect on time, and great talk.